Okay, good day everyone. Welcome to Grade 12 Life Science Revision. So today we'll be looking at responding to the environment with regards to plants. This is also part of Knowledge Strand 2, which is life processes in plants and animals. And the topic that we'll be looking at today is a part of paper 1, and its mark composition is 14 marks out of the 150 marks that we find in paper 1. You guys also know that these different cognitive level questions that we find in each paper. Both paper 1 and paper 2 have different levels of questioning. So 40% would be with regards to knowing science, and then we have 25% which is allocated to understanding science, then 20% is with regards to applying scientific knowledge. Then we also have the remaining 15%, which is for evaluating, analyzing, as well as synthesizing. All right. Just quick study tips. That's very important that you have good time management. Please do ask for help where you do not understand. If there's any topics, concepts, phenomenons that you come across and you seem not to understand them, please do ask for help. Again, practice makes perfect. Use past papers, do activities in your textbook, and that will also just help you to basically measure where you're at and also measure your level of understanding with regards to a certain topic. Then get enough exercise, eat a healthy diet, drink plenty of water, and importantly, do not panic, especially when you're about to go right. Please do not panic. Right. Okay, so let's look at the learning outcomes and summary of today's presentation. So we're going to look at plant hormones, which are auxins, gibberellins, as well as abscisic acid. So we do need to know all these three hormones, and we need to know their functions in plants. Then we're also going to look at the use of plant hormones for weed control. And we're going to look at geotropism as well as phototropism. I believe you guys know that tropisms is a slow movement of a plant. Right, and then we're also going to look at the plant defense. So what we already know is a typical diagram or shoot of a plant. Right, so this was covered in grade 10. We know right here in this structure here, so we have a shoot system, that is the upper part of this plant, and then we also have a root system. Okay, so we're just going to start with the shoot system. So we have an apical bud, which is also called the terminal bud. We have a blade, a leaf blade, right? And then just from the blade there, okay, so from the blade there we have the petiole, okay, and then we have a node, which is just present right there. Then an internode, right, which is present between two respective nodes. Then we also have an auxiliary bud, which is present right there, and this is called the stem. Okay, so now when we get into the root system, we have our lateral root. So these are like your branching roots, the lateral root. So there's one lateral root, the other lateral root, the other lateral root, and the primary root, which is present right there. So guys, it is important for you to know this basic structure of a plant before actually understanding fully what goes on in this topic, right? Okay, so now let's look at these various plant hormones. Okay, so now plant hormones, which are also called plant growth regulators or plant growth substances, they bring about controlled and coordinated growth. They are organic chemicals that occur naturally at low concentrations, very important. And plant hormones are not produced and secreted by glands. We can compare this to, as we had already covered, the endocrine system in humans or in animals, that hormones in animals are produced by glands, but this is not the case in plants. Okay, so now these hormones are not true hormones because they work in the part of the plant where they are produced. So you'd remember that in human beings, the glands would secrete the hormone, and the hormone would travel via the blood to its target organ, right? But in the plant, the hormones work where they are produced. Right. They regulate a plant growth and development from seed formation, germination, through aging and death. So in all steps of plant life, these hormones work there. All right. So during seed formation, you get the plant hormones being present during germination, during aging, even during death. Okay, so now examples, as mentioned, it is our auxins, gibberellins, abscisic acid. We also have another one which is ethylene, but we're only going to focus on auxins, gibberellins, as well as abscisic acid. Right, so let's start off with the auxins. So auxins are created by the shoot apical meristem at the top of the plant. Okay, important. Know that it is created by the shoot apical meristem at the top of the plant. Then the primary role of auxins is to promote cell elongation. So the role or the function of auxins is to promote cell elongation. It also stimulates cell division, plays a role in seed as well as fruit development, and it stimulates root development. It also delays leaf senescence. And here we have a figure 9.3, which shows us shoot, which is treated with rooting hormone, are stimulated to produce new roots. Okay. So in this figure, which is figure 9.2, it says in strawberries, the seeds are the source of auxins. And then we have figure A, which is the normal development, figure B, which is where we have the seeds being removed. And on figure C here, the seeds are removed and auxins are applied. So what basically this figure 9, what it shows us is the development of fruit in strawberries. Okay, so now the seeds of the strawberries, we know that they sit on the outside of the fruit. So now if the seeds are removed before the fruit could swell up, then no fruit development would occur, as we can see here in figure B. But now if a seedless strawberry is treated with auxins, remember auxins is the plant hormone, yeah? Then it will develop as shown here in figure C, right? And it will basically just result in the fruit of the same size as the normal fruit that we have. So it will just be likened to a normal development. Okay, then we have more information with regards to the plant hormone auxins. They bring about a phenomenon which is known as apical dominance. Okay, so in apical dominance, we know that when the shoot apex or the terminal shoot is growing, it usually inhibits development of the lateral buds on the stem beneath, and then the apical buds are found at the leading end of the main shoot. Then the lateral buds are found down the sides of the stems between the leaf stalk and the stem. Then we have a high concentration of auxins, which would inhibit the development of the lateral buds. Then lateral branches would lower down on the main stem and will often grow longer than the lateral branches, okay, which go higher up, right? So now this phenomenon, it gives certain plants their classical triangular shape. We also know that sunlight can reach more leaves, and therefore the plant can maximize on the rate of photosynthesis that takes place. Now, if the terminal shoot of a plant is removed, the inhibition is lifted and the lateral buds can begin growing. Again, this phenomenon of apical dominance is used by gardeners when pruning ornamental shrubs as well as fruit trees. Okay, so we have here figure 2.6.3, where fruit trees need to be short enough to be pruned easily. Okay, now here's the effect again of removing the terminal bud. By figure A, figure 9.4a, auxins are produced by the apical bud and inhibit the growth of the lateral buds. Again, so we have the terminal shoot there, and then the lateral bud growth is inhibited. Ne? Then on figure B, when the apical bud is removed, the lateral buds start to develop. So this is an example of pruning. So once this terminal, or rather the apical bud is removed, then we have lateral buds which start to develop, okay, causing the plant to be, or rather to take up a triangular shape. Okay. Right, so then we move on to the next hormone, which is our gibberellins. Okay, so now gibberellins, they have a stimulating effect on plants. Right, so gibberellins have a stimulating effect on plants, and they basically cause the lengthening of internodes. So remember, internode is found between your respective two nodes, right? So this form of growth is also called bolting. So bolting is basically the growth of an elongated stalk with flowers, right? Okay, then it also causes seeds to start developing only when the conditions they need for successful growth are present. Right, and then gibberellins promote the flowering and delay the aging of leaves as well as fruits. Okay, so here we have figure 9.5. So the plants on the right were treated with gibberellins. So it's a comparison between a plant which was not treated by the hormone gibberellins with the one treated with the hormone gibberellins. So figure B was the one that was treated with a gibberellin hormone, right? Okay, then we move on to the next hormone, which is abscisic acid, and we know it as a growth inhibitor. Right, so abscisic acid suppresses the ability of a plant to grow and develop. Okay, so now this would basically be the opposite of the gibberellins, which is the hormone that we have just covered, right? Because the gibberellins stimulate growth and development in plants. Then abscisic acid is produced in the chloroplast of leaves, and it is responsible for promoting dormancy. Okay, it's responsible for promoting dormancy in plants during winter, and it also delays the germination of seeds. Again, it is involved in the closure of the stomata in times of water shortage. So remember, when the stomata closes, then we avoid water loss from the plant. Then the more water stressed a plant becomes, or the lower the external temperature drops, the more abscisic acid is produced. Okay. So very important, guys, to note the function of each hormone that is covered and to basically know where, or rather what it does in a particular part of the plant. Okay. We'll continue with abscisic acid. It inhibits the opening of buds until a suitable day length has been reached. So this prevents buds from sprouting too early or being damaged by cold weather. Abscisic acid is responsible for forming tough scale leaves. It also delays the ripening of fruit. And then unripe fruit is hard and bitter and will therefore not be readily eaten by animals. Right. So here on figure 9.6, we see high levels of abscisic acid, which prevent the buds of plants from growing, right? And we have bud scales forming right there. Okay. And then weed control. So weed is any plant that grows where it is not wanted. Plant hormones are used to alter the metabolism of weeds in a way that is harmful to them. Then the most widely used weed killers are synthetic auxins. Then one of the benefits of synthetic hormone weed killers is that they are selective. So the hormone weed killer, or rather the weed killer that is used in terms of hormone, it's the auxins. All right. And then the benefits of using these synthetic hormone weed killers is that they are selective as to which weed plants they kill. Right, and then a disadvantage of a selective weed killer is the potential loss of biodiversity in plants that could result from the use of these synthetic hormones. Right, so we have here weed killers which are useful for getting rid of weeds that grow through paving, just as indicated by the figure 9.7. Then on figure 150, we have auxin-based herbicides which are used to control weeds on the roadside. Okay. Right, here we have a summary basically of the hormones as well as the function. And yeah, we can just quickly go through them. So we have the auxins, and a typical auxin that we know of is indole acetic acid, or IAA. So if you guys just know it as IAA, that is also fine. And it is produced, or rather, auxins are produced in the meristems, in shoots, as well as root tips. It acts on young cells in the tips of the plant. And function, it causes young cells to elongate by taking in lots of water. This causes the vacuoles to fill up and elongate the cell, while the cellulose cell walls are still elastic and can stretch. Then when we look at the effect on the plant is apical dominance. So we have high concentration in the apical bud, which stimulates growth. Then we also have high concentration in the lateral side, or the lateral buds, and that stops growth. Right, so further away from the apical bud, the lower concentration of the auxin stimulates growth. Okay, then young leaves, the auxins are produced in young leaves, and it would act on the leaves. So it would cause a special layer of cells called the abscission layer at the base of the leaf stalk, and this would result in the falling of leaves and or of fruit. Okay, so the effect on the plant is that the leaf or the fruit would fall. Then as leaves age, or when there is a decrease in temperature, the cells of the abscission layer become waterproof like cork. Then the leaves no longer receive water and they die. So the leaves fall off the plant, and this is called abscission. So abscission is basically dropping off. Okay. Then we also have auxins, which are produced in seeds in the ovary. Okay, and they would act in the ovary of plants. Okay, so the auxins would cause the ovary to swell and grow, forming the fruit. Okay, so then the effect on the plant would obviously be fruit development. Then we also have auxins, which are produced in the cells of the stem, and they basically act on the cells in the end of the stem, for example, a cutting. The function basically of these auxins, which are produced at the stem, is to stimulate the cells of the stem to grow adventitious roots. Okay, so the effect on the plant is root formation. Then we know that cutting of stems are dipped into a powder containing synthetic auxin, and it stimulates growth of the adventitious roots, just like the picture that we had seen. Okay, then the next hormone, abscisic acid. So abscisic acid would act on leaves and shoots, and it functions on leaves and shoots would be growth inhibiting hormone. So remember we said that abscisic acid is a growth inhibitor. Right, so it allows plants to respond to stress, for example, drought or frost. And then its effect on the plant is that during autumn, leaves of some plants produce abscisic acid, which stops the growth of tips of shoots and lateral branches. And it also causes buds to become dormant. Then when abscisic acid acts on seed, we have dormancy of seed. So it basically prevents germination. And then the effect on the plant is that it gives the seeds time to mature and to only be dispersed when they are ready to germinate. So not all seeds will germinate at the same time. Germination can only happen when the amount of abscisic acid decreases, right? So this would usually happen when spring rain washes away the abscisic acid as the seed passes through the gut of an animal. Because now abscisic acid can also act on roots, and on roots, basically its function is under stress when there is too little water. Then the effect on the plant is that it is then carried to the leaves, where it causes the stomata to close, right, in order to prevent water loss, right? Then the gibberellins, they cause rapid elongation of cells. Okay, so the effect on plant is that we have the growth of roots, we have an elongation of stems, we have the bolting in rosette plants, for example, the lettuce as well as the cabbage. Right, let's move on. Okay, so we have a question. It says, auxin-based selective herbicides, A, are made using auxins, B, only kill weeds if they are used in the correct dosage, C, both A and B, or is it D, neither A nor A? No, B, sorry. Auxin-based selective herbicides, A, are made using auxins, B, only kill weeds if they are used in the correct dosage, or C, both A and B, or is it option D, neither A nor B? Okay, so remember, for multiple choice questions, you can always use the elimination method. So if you know what auxin herbicides or selective herbicides are, then it is very much easy to note the answer in this case. So we know that auxin-based selective herbicides are made of auxins because already the title tells us that. And they only kill weeds if they are used in correct dosage. That would also be very true. So our answer is C, which is both A and B. Okay, next question. The advantage of auxin-based herbicides is or are A, no need to weed, B, they are pet and children friendly, or is it C, reduces labor because there is no need to weed? Is it A, no need to weed, B, they are pet and children friendly, or is it C, reduce labor because there is no need to weed, or D, all of the above? And guys, if you say D, that is correct. Okay, and let's move on to the next question. It says, in roots, a high concentration of auxins, A, promotes growth, B, inhibits growth, or is it C, both A and B, or D, none of the above? In roots, a high concentration of auxins, A, promotes growth, B, inhibits growth, or C, both A and B, or D, none of the above? Okay, and the answer is B, it inhibits growth. Okay, so remember, this is in roots and not in stems. Okay, so now we move on to tropisms in plants. Remember, I said that tropism is a slow plant movement. Okay, so tropism refers to slow plant movement, and it is a growth response of the plant to an external stimulus. Then the stimulus could be gravity, could be light, or even the presence of water. Right, so a negative response is growth away from the stimulus. So we would say it is a negative tropism. And then a positive response is growth toward the stimulus. And we would also say that is a positive tropism. Okay. So for example, if a plant grows toward gravity, if gravity in this case would be the stimulus, then we would say that it is positive tropism. And then if it grows away from the gravity, if gravity is the stimulus, then it would be a negative tropism. Right, so tropism is brought about by the hormone auxins. Okay, very much important to note that tropisms is brought about the auxins, and then the direction of the tropism is dependent on the direction of the stimulus. Okay, so the direction of the tropism or the direction of the plant growth is dependent on the direction of the stimulus. Okay. And then these auxins are found at the tip of roots as well as shoots. Then we have the IAA, which is the most well-known auxin, or indole acetic acid. Right, so now we're going to look at two types of tropism, which is geotropism as well as phototropism. Right, let's start off with geotropism. So geotropism is a bending reaction in plants to the stimulus of gravity. right? And then let's look at the role of auxins during geotropism. So in this case, the stimulus would be gravity, right? So in the root, auxins are produced at the tip of the root. So now if the auxins are evenly distributed around the root, then the root grows straight downwards. Then if a root is placed horizontally, gravity pulls the auxins to the lower side of the roots. Therefore, there is an uneven distribution of auxins. Then one side would have more auxins than the other side. And in this case, in roots, a high concentration of auxins would inhibit growth. Therefore, the upper side with less auxins will grow faster, and the upper side will therefore bend towards gravity. Right. So now, since the roots grow towards gravity, we say that it is positively geotrophic. Right. So here is basically a diagrammatical explanation of what we have just seen. So the first diagram, it says it's showing us geotropism in a young seedling. Okay, so we have a radicle, which is from the newly germinated seed, and we also have a shoot. Okay, so now this is what happens when geotropism takes place. So the radicle is positively geotropic, it meaning that it moves down, right? Moves in the direction of gravity. And then we have the shoot, which is negatively geotropic, right? Meaning that it moves away from the stimulus of gravity. Okay, so you guys need to be able to just draw a simple diagrammatical explanation or picture rather of geotropism. Right, so then the next diagram is showing positive geotropism in roots. Okay, so here we have a root tip which produces auxins. So the auxin is present right there. Then less auxin is on the upper side, right? Meaning that the auxins are moving towards this side, they are moving down. Yeah? And then now the lower side would cause more elongation in the root. So remember, these arrows are showing us the movement of the auxins. The auxins are moving down, meaning that this side would elongate, causing the root to bend, right? So we have higher auxin concentration, which causes less elongation in the roots on this side, but we have more elongation on the upper side, right? And when we look at the diagram at the bottom, it shows suggested explanation for negative geotropism in shoots. Okay, so we'd have less auxins on the upper side and we'd have more auxins on the lower side, right? And this is what would happen. The lower auxin concentration would cause less elongation in the shoots, and then where there's higher auxin, it would cause more elongation in the shoots, causing it to bend upwards or just basically grow in an upward position. And this would be negative geotropic, right? Then we move on to phototropism. Right, so phototropism is basically a response of plants to the stimulus of light, right? Then the role of auxins in phototropism is that auxins are produced at the tips of the stem. Then they move down evenly and cause even growth. They ensure the stem grows upright. And sometimes the auxins are exposed to light from one side only, and we call this unilateral light or unidirectional light. Then auxins, we know that they are destroyed by light. Therefore, we find that the side exposed to unilateral light has less auxins. So the side which is exposed to unilateral light or unidirectional light would have less auxins. Okay, now this is because the auxins are destroyed by the light or they move to the darker side or the shaded side, right? So now the role of auxins in phototropism is that in stems, more auxins promotes growth. So therefore, the darker side will grow faster than the lighter side. So when we speak of the darker side and the lighter side, the darker side is the side which is not exposed to too much light. Then the lighter side is the side which is exposed to the light. Then this is because the darker side has more auxins. Remember, auxins are destroyed by light. They will try to move to a side of the plant which is shaded. So now, since the darker side grows faster, it causes the stem to grow towards the light. Therefore, we can say that the stems are positively phototrophic because they grow towards light. Okay, so here we have an example. So we have light here, which is distributed evenly upon a plant. So we have a seedling in even light, right? Now we can also see the straight or upward growth of this plant. Okay, then in figure B, we have the seedling, which is in unidirectional light, or what we call unilateral light. And we see that there is a bending reaction of the stem or the plant towards the light, or rather towards the direction of the light. So remember in the previous slide, we did mention that plant movements depend on the direction of the stimulus. Okay. Okay, so here we can also see the auxin activity in phototropism as to why this seedling in unidirectional light bends. Okay, so firstly, we know that auxins are produced in the tip of the stem, and this is our light. This is where our stimulus comes from. It's a unidirectional light or unilateral light. Right. So now when this stem is exposed to this unidirectional light, the auxins would move down to the dark side of the stem or the shaded side of the stem, and they would cause cell elongation on this darker side or the shaded side. And then on the light side, or rather the side of the stem where light is exposed, the cells will not elongate, so the stem would bend towards the light or bend towards the stimulus, right? So that is basically the auxin activity in phototropism. Right, let's look at investigations to show tropisms. So a clinostat is a special piece of laboratory apparatus that is used to study tropism, and here we have the clinostat. We also have the rotating disc. So inside the clinostat, we have P seedlings with horizontal radicals, and we also have a wide-mouthed jar, right? And the stationary cork disc, which is attached to the clinostat. Okay, so firstly, the wide mouth glass jar is attached to the rotating disc and then to the clinostat. Okay, so now there is a motor in the base which turns the disc around. Now it is difficult to eliminate the force of gravity, but rotating the seedlings on the clinostat, the force of gravity is then neutralized. Then as the disc revolves, each side of a seedling is influenced by the force of gravity, right? So here we have a summary which is tabulated and also showing the different roles of auxins in phototropism as well as in geotropism. So remember, in phototropism the stimulus is light, and then in geotropism the stimulus is gravity. Okay, so we can just go through it quickly. Okay, so in phototropism, auxins are produced at the tip of the stem or the shoot, but then in geotropism, it is produced at the tip of roots. Okay, then auxins would move down evenly during phototropism and even during geotropism, but they would move upwards. Okay, so in phototropism, they move downwards. In geotropism, they move upwards. Okay, then again in phototropism, this even distribution brings about equal growth on all sides of the stem. So this is when we have even light. And then when we go back to geotropism, this even distribution brings about equal growth on all sides of the root. Okay. Then we go back to phototropism. As a result, the stem would grow upwards. And then when we go to geotropism, the roots would grow downwards. Okay. Back to phototropism, when the stem is exposed to unilateral light. So remember, unilateral is also unidirectional, that is light from only one side. The auxin concentration will be high on the dark side or the shaded side because light destroys auxins. Right. Then when you go to geotropism, when the root is placed horizontally, so this would be an example of the germinating seed. Only one side is exposed to gravity, then the auxin concentration will be high on the lower side of the root, and gravity then attracts the auxins. Okay, back to phototropism. More growth occurs on the dark side because auxins stimulate growth on the dark side. Go to geotropism. More growth occurs on the upper side of the root because auxins on the lower side inhibit growth. Then we have in phototropism the stem bending towards the stimulus or towards the light. Then in geotropism we have the root bending downwards, okay, or towards the stimulus of gravity. Right. Question: when the root bends towards gravity, this is known as A, positive geotropism, B, negative geotropism, C, positive phototropism, or is it D, A, and C? When the root bends towards gravity, this is known as A, positive geotropism, B, negative geotropism, or C, positive phototropism, or D, both A and C. Okay, so now what we should first note in this question is the stimulus that we find. Okay, so we know that the question is about a tropism because we have a plant movement, but now the stimulus that we are asked about in this question is gravity. Okay, therefore, meaning that we are talking about geotropism. So we are already cancelling out option C and we are cancelling out option D because option D also has option C, and we know that phototropism does not involve gravity. Okay, so now we are left with option A and option B. We know that roots are positive geotrophic towards gravity, right? So therefore, our answer would be A, right, which is positive geotropism, right? Okay, next question. When the stem grows towards the sun, this is known as A, positive geotropism, B, negative geotropism, C, positive phototropism, or D, both A and C. Again, we know that we are dealing with a tropism or plant movement. And when we're dealing with plant movements, we need to know what the stimulus is. And in this case, or in this question, the stimulus is the sun. And we know that the sun gives light, right? So meaning that we are dealing with phototropism. And here our only option for phototropism is option C. Okay. Then we can move on to the next question, which says roots and stems grow evenly if there is A, an even distribution of auxins, B, an uneven distribution of auxins, or C, an uneven distribution of gibberellins, or is it D, none of the above? So we are looking for even growth. Right, so we know that we need to have an even distribution of auxins. Okay, so when you have an even distribution of auxins, which is present in roots, then our root would grow evenly downwards. And if we have an even distribution of auxins in stems, we know that our stem would grow evenly upwards. Okay, then we move on to plant defense mechanisms. So defense mechanism is required by plants to protect themselves against attacks by herbivores and as well from pathogens. Okay, so some examples of pathogens are fungi, bacteria, as well as viruses. So plants are able to protect themselves using chemicals or thorns. So it either uses a chemical method or a mechanical method, right? Okay, so let's look at thorns, which is a mechanical way for a plant to protect itself. So thorns may be found on leaves and stems of some plants. They are evolutionary developments. Some examples of plants is that they have thorns are cacti, roses, as well as lemons. Functions of these thorns is basically to protect the plant from herbivores, and when the plant grows in bunches, then the thorns work together to bring about a group defense. Okay, so thorns also help to disperse the plants over wide areas. Then when we look at the chemical defense, or rather the chemical plant defense mechanism, is that some plants secrete chemicals. Sometimes these chemicals can be poisonous to other organisms. One example is the high vitamin C and high bioflavonoid content of some plants. And these chemicals have antibacterial and antiviral components, meaning that these chemicals are harmful to bacteria and viruses. And then other plants have sticky secretions, and the sticky secretions make it difficult for insects and animals to eat the plant. Then some chemicals are used as chemical messengers between plants. Then we also have these chemicals releasing an odor. Then other plants will detect this odor and can then strengthen their defense mechanism. Here's an example of a plant that does this is the sage bush. Then now when the bush is attacked by insects, it releases a chemical called methyl jasmonate. And then other plants will detect this chemical and increase the levels of a certain enzyme, which is called PPO. So PPO would prevent leaves from being attacked by other parasites. Okay, so now other plants would secrete a chemical that attracts insects that feed on the larva of the pest or the parasite. As an example, we have the monarch butterfly, which feeds on the larva of the milkweed. And here we have in figure 9.16 a drop of resin on a pine tree trunk. So that is the sticky secretion which is released by the plant. Then figure 9.19, it's a micrograph of the irritant hairs on the surface of the stinging nettle leaf. Then we have figure 9.22, where the giraffe is using its long, flexible tongue to pick off leaves from between thorns. Okay, so remember the thorns are there to protect the plant from herbivores. Okay, then we have the cactus plants right there. Okay. Okay, question. So the enzyme that is responsible to protect the leaves against the attack of parasites is called A, PPO, B, methyl jasmonate, C, both A and B, or is it option D, neither A nor B? The enzyme that is responsible to protect the leaves against the attack of the parasites is called A, PPO. Is it option B, methyl jasmonate? Or option C, both A and B? Or is it option D, which is neither A nor B? Okay, so we have two options basically, which is PPO and methyl jasmonate, and we need to establish basically which one is the enzyme. So methyl jasmonate is a chemical which is secreted by a plant which is in danger, right? And the surrounding plants would then release the enzyme PPO to protect themselves. Right, so now the enzyme that we are looking for is option A. Right, next question. Plant structure that is able to protect the plant against attack from herbivores is or are A, a sticky secretion, B, thorns, C, both A and B, or is it D, neither A nor B? A plant structure that is able to protect the plant against, or rather against attack from herbivores. Is it A, sticky secretions? Is it B, thorns? Or is it C, both A and B? Or is it option D, which is neither A nor B? Okay, so firstly, when we look at the question, we are looking for a structure and not a secretion, and immediately it eliminates option A and we are left with option B. So our answer in this question would be thorns. Against sticky secretion usually works against insects and other small parasites. Right. Okay, so here we have basically, guys, just a summary of what we have learned in this topic of responding to the environment with regards to plants. Okay, so we have went through plant hormones. We went through weed control by using growth hormones, which was auxin, right? An auxin selective weed killer. And then we also went through the general functions of auxins, gibberellins, as well as abscisic acid. So very much important to note these functions of auxins, gibberellins, as well as abscisic acid. Okay. Then we also looked at the different tropisms that exist in plants. We looked at geotropism, which is the effect of gravity on the plant movement, and we also looked at phototropism, which is an effect of light on plant movement. So, and it is regulated by auxins, right? So you guys should know how these auxins go about in order for geotropism to take place as well as phototropism to take place. Again, remember we have negative as well as positive tropisms. So if it's a negative tropism, it means that the plant moves away from the stimulus. And if it's a positive tropism, it means that the plant grows or moves towards the stimulus. Right. So we also looked at a plant defense mechanisms. We looked at the several chemicals which are released by plants to basically defend themselves from predators. And we also looked at mechanical ways, which would be using thorns, or rather the plants having thorns. Okay. Right, now we can move on to the terminology that we have covered in this topic. Again, guys, make mobile notes or flashcards with regards to terminology of every topic. It will also help you to basically master this topic and make you mobile in your studying. Right, so the first definition that we looked at, or rather the first term that we used during this topic, was hormone. We know that it's a chemical substance which is produced by an organism to control and regulate cell activity in that organism. We also looked at apical dominance, which is the inhibition of the growth of lateral buds by the presence of a terminal bud on the main stem. Then we also looked at internode, that's a section of stem between two nodes. We looked at bolting, which is the growth of an elongated stalk with flowers. We looked at dormancy, which is the period of inactivity. Then carcinogenic causes cancer. Then we also have tropism. This is growth response of a plant to an external stimulus. We looked at geotropism, which is a plant response to the direction of gravitational pull. And then phototropism is growth movement of a plant in response to light. Then a pathogen is a disease-causing organism. Toxic, this means poisonous. Then transpiration is loss of water vapor from the aerial parts of the plant, especially the leaves, via the stomata. Right. And then we can get to exam questions. So guys, just before we get into the questions, please do pause the video once the questions are revealed so that you just answer the questions alone first, and then we can go through your answers, or rather you can compare your answers with the answers that I have. All right. Okay, so the first question that we have is question 3.2. And it reads, auxins influence the growth of stems and roots. Okay. Then question 3.2.1, state where auxins are produced in a plant. And then question 3.2.2, describe the effect of auxins on a plant stem that is exposed to unilateral light. Okay, so let's start off with question 3.2.1, which is only 1 mark. Where are auxins produced in a plant? Okay, so in the tip of the stem, tip of the root, the apical meristem, terminal bud or apical bud, and you would get your 1 mark if you say either, right? Then when we move on to question 3.2.2, describe the effect of auxins on a plant stem that is exposed to unilateral light. Okay, so firstly, auxins are produced in the tip of the stem. You mentioned that you get your 1 mark. Then when exposed to unilateral light, the auxins move down to the darker side of the stem, causing the cell to elongate on the darker side. then the stem would then bend towards the light. Right, so if you mentioned where the auxins are produced, and if you also mentioned the direction of the auxins once exposed to the unilateral light, you would get your second mark. And if you also mentioned where the cell elongation takes place, you would get your third mark. And if you also mentioned then the physiological reaction of the plant, you would get your fourth mark. Okay, so we know that the plant would bend towards the light. Okay, next question, which is question 3. It says the diagram below represents a germinating seed. Assume that the radicle and the pumule were exposed to uniform light from all directions. Okay, then question 3.1.1 says define tropism. Then question 3.1.2, draw a labeled diagram to show the position of the radical and the pumule in the seedling 1 week later, right? Okay, so we have the radical, which is your young root, and we have your pumule, which is your young stem, right? So question 3.1.1 says define tropism. Tropism, we know that is growth of a plant in response to a stimulus, or if you said it's slow plant movement or growth towards stimulus, that would also be correct for your 2 marks. So keywords that we're looking for here as stimulus, as well as growth or movement of a plant. Okay. Then question 3.1.2, draw a labeled diagram to show the position of the radical and the pumule in the seedling 1 week later. So this is a germinating seed, and we're told that it is exposed to uniform light from all directions. Okay. So here we have the picture or the diagram that we had to label. Firstly, your radical, which is a young root, we know that it is negative. It is positive geotropism, has positive geotropism, meaning that it will need to go down or grow downwards, right? And then the pumule, which is our young stem, is positively phototropic, meaning that it will need to go up, right? Then our germinating seed will obviously be there. Then what you need to do basically is to label. So if your pumule is going up, then you'd get your 1 mark. If your radical is going down, then you'd get your second mark. Remember, it is 4 marks. And then your third mark would go for your caption, which is seedling after 1 week. And then your fourth mark would go for any other correct label that you have put in your diagram. Right, so in this case, if you just labeled this part and said a germinating seed, you would get your 4th mark. Right. Then we move on to the next question, which is also part of question 3. It says the diagrams below represents the growth responses of 2 different plant organs to external stimuli. Okay, so if we look at the diagram, we have A, which is, we see the direction is from that side, and we have this part B, or the plant is B, and plant B, once it gets the stimulus of A, it bends towards A, and then plant C, here, we see that its direction is downwards to the stimulus at D, right, or to the external stimuli of D. So these are different plant organs, organ B as well as organ C. Okay, so then let's go through the question. Question 3.1.1, name the group of plant hormones that is responsible for the growth responses observed in the diagrams. Then question 3.1.2, name the external stimulus at A as well as at D. Then question 3.1.3, give one observable reason why a plant organ B is a stem. Question 3.1.4, explain the growth response observed in plant organ C. Okay, then we can go back to question 1. Name the group of plant hormones that is responsible for the growth responses observed in the diagrams, and it is auxins. We know that auxins bring about tropisms. Okay, then we move on to question 3.1.2. Name the external stimulus at A as well as the external stimulus at D. So that is for question 3.1.2b. Okay, so at A, we are supposed to name the external stimulus, but because, guys, we had already read through all our questions, if we just go to question 3.1.3, it says give one observable reason why plant organ B is a stem. And we see that the stem is bending towards the stimulus A. 1 plus 1 is 2, meaning that stimulus A would be light, right? So this is if you do not know or you are confused or you forgot, right? So you can, it's also just a helpful technique to just first read through all your questions and then go back to the first question or to the description of the question and then just do it, or rather do the question then, right? Just to get an overview of what you are asked and what you should be looking for. Okay, then question 3.1.2b. We're supposed to name the external stimulus at D, and that would be gravity, right? In question 3.1.3, give one observable reason why a plant organ B is a stem. So the observable reason that we see there is that the plant structure has bent towards the light. And then question 3.1.4, explain the growth response observed in plant C. Explain the growth response observed in plant C, and that is for 3 marks. So basically, we are explaining the auxin activity during geotropism. So auxins accumulate on the lower side of the root. Then high concentration of auxins on the lower side of the root inhibits growth. Then the lower concentration of auxins on the upper side stimulates growth, causing uneven growth, or what we call positive geotropism. Okay, so that is for 3 marks. So if you mentioned any 3 points, then you get your 3 marks right then. Okay, next question. The diagram below shows the results of an investigation carried out to determine the effect of auxins on the growth of cleopetals, which are young shoots. So the procedure was as follows. 3 cleopetals were used. Cleopetal A, which is this cleopetal here, had its apical bud removed. Then Cleopetal B had its apical bud covered with a black plastic cover. Then Cleopetal C had its apical bud covered with transparent plastic cover. Then we see that all 3 Cleopetals, which is A, B, and C, were then exposed to unilateral light or sunlight. Then the diagram below shows the results of the investigation. So the unilateral light or sunlight came from this direction here, rather from the left. Okay, go through the questions. Question 2.5.1, explain the results obtained as shown by Cleopetal B and Cleopetal C. And in question 2.5.2, explain why lateral branches will develop in Cleopetal A. So let's start with question 2.5.1a, where we need to explain the results of Cleopetal B. Because in Cleopetal B, we see that it is just up straight. There is no bending towards the unilateral light, and our mark allocation there is 3 marks. So we need to mention 3 points. Okay, so the shoot grows straight up and you get your first mark. If you mention that the tip of the shoot does not receive any light, you would get your second mark. And if you also say that the auxins remain evenly distributed in the tip, you would get your third marks. And also all parts of the shoot are equally stimulated to grow, you would get your next mark. Right, and then when we move on to question 2.5.1b, where we need to explain results for, or rather that are observed by Cleopetal C, which is the cleopetal, which was covered in transparent plastic cover. And we also see a bending of the cleopetal or the shoot there, right? We see a bending towards the unilateral sunlight. Okay, so the shoot bends towards the light. You mentioned that you get your first mark, and then because it is exposed to unilateral light, you mention why it's bending, you get your second mark. The auxins in the tip move away from the lighted to the darker side of the shoots. Then the cells on the darker side are then stimulated to elongate. Growth is inhibited on the lighted side. So you would get your 4 marks there. Any 4 points which I mentioned, you get your 4 marks. Okay, then question 2.5.2. Explain why lateral branches will develop in cleopetal A. Why would we have lateral branches developing in cleopetal A? So since the apical bud was removed, no auxins are produced, therefore there is no apical dominance. Okay, so it's because we don't have the phenomenon of apical dominance. Right, next question, which is question 2.4. It reads as follows. A grade 12 learner performed an investigation to determine the effect of light on the growth of plant shoots. The learner divided the plants that were used into 3 groups as follows. We have group A. The tip of the shoot was intact. So group A is this one. That was the start and that was the end. And then group B, which is in the middle, the tip of the shoot was removed. So as we can see, there is no tip there. Then group C, the tip of the shoot was covered by a cap that does not allow light to pass through. Because another diagram below shows each shoot at the start of the investigation and next to each the same shoot at the end of the investigation. So now the arrows indicate the direction in which in each investigation, right? Then we have 5 questions there. Question 2.4.1 says name the dependent variable in this investigation. Question 2.2 says state 2 factors that must be kept constant in this investigation. Question 2.4.3, the influence of which plant hormone is being investigated. Then question 2.4.4, explain the results observed in investigation A and C as illustrated in the diagram above for 6 marks. Then question 2.4.5, state 2 ways in which the learner could improve the reliability of this investigation. Okay, and now we can start with answering our question. We know that we're dealing with investigation type of questions where we need to basically identify our dependent variable, independent variable, look at the aim of the investigation, and also look at the conclusion as well of the investigation, look at the method that was used, look at the apparatus that was used, and just keep that in mind. All right, question 2.4.1, name the dependent variable in this investigation. So the dependent variable, guys, remember it is the variable that is measured. Right, so in this case, we are measuring the growth of the plant shoots. Okay, then question 2.4.2 says, state 2 factors that must be kept constant in this investigation. 2 factors that must be kept constant in this investigation. So we need to put the plants, right, in the same environment, or the shoots in the same environment, and we need to use the same type of shoot, okay, in order to make our investigation reliable. Okay. Then, question 2.4.3, the influence of which plant hormone is being investigated. We know that auxins, they play a role in tropisms, so it would be auxins. Then question 2.4.4, explain the results observed in investigation A and C as illustrated in the diagram above. Okay, so we can start off with investigation A. Ne? So investigation A, the tip of the shoot was intact, right? And it started off in a straight, it's an upward growth, but then at the end of the investigation, it grew towards the stimulus, right? So we see that the stimulus came from this particular direction there, right? So let's start off with investigation A. So light from the right side caused the auxins to move to the shaded side of the shoot, leading to increased cell elongation as well as cell division, right? And then there was therefore greater growth on the shaded side, thus bending the shoot in the direction of the source of light or of the stimulus. And then when you go to investigation C, light, this is investigation C, where the tip of the shoot was covered by a cap that does not allow light to pass through, right? And it's also has a unidirectional light. So light has no influence on the distribution of auxins. Therefore, the shoot grew upright, right? So for investigation C, you would get 2 marks, and for mentioning what happened in investigation A, you are allocated 4 marks. All right, so any 4 points mentioned in investigation A, you get them, and then any 2 points mentioned in investigation C, you get your full 6 marks. Okay, then we move on to question 2.4.5. State 2 ways in which the learner could improve the reliability of the investigation. Guys, if you find this question in any with regards to any investigation, repeating the investigation gives is always a way to improve the reliability. All right, so seeing basically just measuring the results or comparing the results that you got from your first experiment or your first investigation, you compare them to your second one, you compare them to your third, so on and so forth. So repeating an experiment or repeating an investigation basically improves or increases the reliability of your findings, right? So repeat the investigation. Then another way in which we could improve the reliability of this particular investigation is to use more than one plant for each treatment, right? So meaning that we use a particular plant for the first time we do the investigation, and the second time we would use a different type of plant or different type of shoot, and so on and so forth, to just measure or see if the results or the findings are the same or similar, right? Then we move on to the next question, which is question 3.2, and it reads: botanist investigated the effect that abscisic acid had on the germination of seeds. So the procedure was as followed. He placed equal numbers of lettuce seeds in 2 potting trays, potting trays A and B. Each tray contained the same soil and was subjected to the same environmental conditions throughout the investigations. Already, as you are reading the procedure, you are told what was kept constant. So guys, when reading such questions or when approaching such questions, please have a highlighter, have a colored pen, have a pencil, underline the important factors or things that you are given in these statements. Ne? Then we move on to the second point. The percentage of seeds that had germinated in tray A was calculated every 10 days to 40 days. Then at the same time, a sample of seeds was taken from tray B, and the concentration of hormone in the seeds was determined. Okay, then the results of the investigation are provided in the table below. So 0 days, we had 0% seeds germinated in tray A, then concentration of abscisic acid in seeds in tray B was 350, then so on and so forth. So we have our time there in days and the percentage of seeds that germinated in tray A, as well as in the third column, it is the concentration of abscisic acid in seeds in tray B. Okay. which is measured in nanograms per gram. Okay, question 3.2.1. Formulate a hypothesis for this investigation. Formulate a hypothesis for this investigation. So in order to formulate a hypothesis, you first need to know what the aim of the investigation. So we go back to our description there. It says, and botanists investigated the effect that abscisic acid had on the germination of seeds. Right. So we have abscisic acid and we also have its effect on germination of seeds. So this is a statement of what you could have or what you will see in this investigation, right? So whether it's a positive or a negative hypothesis, either way would be correct. But the important thing about writing a hypothesis is that you need to include both variables, right? So you need to include both your independent variable as well as your dependent variable. And here I have an hypothesis which says abscisic acid has no effect on germination. And if you said that abscisic acid would have a positive effect on germination, have a negative effect on germination, then it would also be correct, right? But the important thing here is to list both your, or rather to include both the independent as well as the dependent variable, right? Then we move on to question 3.2.2. Identify each of the following in this investigation. A, the dependent variable. Dependent variable. And then question 3.2.2, the independent variable. Right, so the dependent variable would be the percentage of seeds germinated, and then our independent variable would be the presence or the absence of abscisic acid. Right. Dependent, the percentage of seeds that germinated, and our independent variable would be the presence or the absence of abscisic acid. Right. So even the answer to question 3.2.2, you can also find it in the first line there, which says a botanist investigated the effect that abscisic had on what? The germination of seeds. So guys, just take your time when reading these descriptions, because most of the answers are found there, right? Especially during investigation and experiment questions. And then question 3.2.3, state two ways in which the botanist ensured the validity of the investigation. So when we talk about validity, we're basically just saying, is this investigation reliable? Are the results or the findings that the botanist got reliable or valid, right? So certain measures need to be taken into place in order for our investigation or results to be reliable. And then the question is asking us, what did the botanist do to ensure that these findings that he got are valid? Again, we go back to what? Our description. We say that he placed equal numbers of lettuce in the two potting trays. Each potting tray contained the same type of soil. So he didn't put clay soil in one tray and sand in the other tray. We had the same type of soil. And the trays were also subjected to the same environmental conditions throughout the percentage, right? So the answer to question 3.2.3, we had equal number of seeds in both trays. We had the same type of soil which was used in both trays, and data was also collected from both trays at the same time. Right, so all of this is mentioned in these description right here. As we can see there by the third point, it said at the same time, a sample of seeds was taken from tray B, and the concentration of the hormone in the seeds was determined. Right. And the first point also tells us about equal numbers. And then we move on to question 3.2.4. What would the concentration of abscisic acid be in the seeds in tray A after 50 days? What would the concentration of abscisic acid in the seeds in tray A be after 50 days? So if you said 0 nanograms per gram, that would be correct. Or if anything that is less than 10. Okay, so where we are referring to this one here. Okay, so if you mention anything less than 10, then you would get your mark. If you said 0, you'd also get your mark because it's lower than 10. Right. Let me move on to the next question, question 3.2. And it reads, a group of grade 12 learners set up the following apparatus in their classroom. Two plants were used. One was placed on a clinostat that rotated. That would be in diagram A. And then the other was placed in a stationary position at diagram B. Okay? They left the apparatus in these positions inside a dark cupboard for 2 weeks before making the observation as indicated in the diagram below. Then it says, note, a clinostat is an apparatus that is able to rotate. Okay, then question 3.2.1. What type of plant growth movement was observed? Question 3.2.2 briefly explain the results observed in A and B. In question 3.2.3, state one reason why the apparatus was placed in a dark cupboard for the duration of the investigation. Right, let's start off with question 3.2.1. What type of plant growth was observed, or plant growth movement, was observed? And that would be tropism, right? And then question 3.2.2 briefly explain the results observed in A as well as B. A, as the plant was constantly rotating, there was equal distribution of auxins throughout the stem, and therefore there was no unequal growth of the stem. Then the stem continued to grow straight and upwards. So that is diagram A. Diagram A, which is right there. And then we move on to question 3.2.2b. A high concentration of auxins on the lower side of the stem stimulated the growth of the stem. Thus, an equal distribution of auxins caused unequal growth of the stem on the lower side, causing the stem to grow upwards. All right. Then we move on to question 3.2.3. State one reason why the apparatus was placed in the dark cupboard for the duration of the investigation. Okay, so this is to eliminate the effect of light. Otherwise, phototropism would have also played a role in this investigation, and that was for 1 mark. Right. Okay, next question. The diagram below shows two plants, A and B. So we have plant A, which is present here, and plant B present right there. Okay, at the start of an investigation, the plants were treated in the following ways. First point, no changes were made to plant A. The apical bud of plant B was removed. Then each plant was covered with a box with a single opening as shown in the diagram and placed in a lit room. Okay, so we see the opening right there. So this would be the box with the opening, and that's the box again with the opening on the side. Then question 3.5.1, state the role of the boxes in this investigation. Would be to ensure unilateral light, because light will only enter in one direction. And question 3.5.2 says, name the hormone that is removed by cutting off the apical bud from plant. Name the hormone that was removed by cutting off the apical bud from plant. By now, you should know that it is auxins, right? Or acid. Then question 3.5.3, tabulate two differences between plants A and B that you would expect after 2 weeks. Right. So here we have our differences. Plant A, the stem of the plant will bend towards the light, And then in plant B, the stem of the plant will remain straight, or it will not bend towards the light. Okay, so in plant B, we have lateral branches which will grow. Then plant A, another thing that we can mention is that the plant will be taller. And then in plant B, the plant will be shorter or would have a triangular shape. Okay, so then mark allocation with regards to question 3.5.3, you get a mark for basically answering in a table form. You get your 1 mark for mentioning the two differences that you'd find in plant A as well as in plant B. So if you have 2 points under each plant, then you get your 4 marks, and then for answering in a table, you'd also get your other mark. Guys, remember, it is important for you guys to write captions when writing or drawing a table or answering in a table format. Sometimes marks are allocated for the caption. So always write a caption for every diagram that you draw, every table that you're asked to draw, and every graph that you are asked to draw. Okay, then we move on to question 3.5.4. The diagram below shows plant B 7 days after being sprayed with gibberellins. Right, and then it says explain the effect that the gibberellins had on the plant by referring to the changes observed in the diagram. Okay. Okay, so the gibberellins cause the stem to grow longer because gibberellins stimulate the elongation of the internodes. Right, so that's all that we needed to mention because that is just 2 marks. Right. So you mentioned the effect that the gibberellins had and why it had that effect. And then we move on to the next question, which is question 2.2. Says a clinostat is a device used to investigate plant growth responses. It has a disc that rotates very slowly when the clinostat is switched on. So during an investigation on plant responses to light, the procedure below was followed. We have 3 pots of plants of the same species we used. Then each pot plant was placed on one of 3 identical clinostats. Each set of the apparatus with A, B, and C was placed in a box with a single opening. And then each clinostat was treated in a different way over a period of 5 weeks. The results of the investigation are represented in the diagram. So we have our plants here, and with the opening, both on, or rather all 3 of the openings are on the right hand side. Okay, then question 2.1 says name the plant growth response to light. Again, that would be the auxins, or rather, sorry, sorry guys, that is the plant growth response, not the hormone. Plant growth response to light. That's phototropism. Okay, then question 2.2. State 2 factors that were kept constant during the investigation. We have the same type of plant species, and then we also have identical clinostats that we used. We have the same period of time which was used for each setup, and then each apparatus was placed in a box with a single opening on the same side. Right. Then question 2.2.3, give one reason why the results of this investigation may be considered to be unreliable. Give one reasons as to why the results of the investigation may be considered to be unreliable. Okay, so the investigation was only done once, it was not repeated, right? And then only 1 plant was used in each setup. Okay. Then we move on to question 2.2.4. In which apparatus A, B, or C was the cleaner A switched on and rotating slowly, and then B switched off and manually rotated through 180 once a week? Because then I would need to look at the way the plant grew. Okay, so the one way we would say that it was switched on and rotating slowly would be plant B. All right, plant B, and then it was switched off but rotated manually through 180 once a week would be plant A, because we can see the spirals that go here on the plant A moves like that. See, it's the growth pattern of the plant. Okay. Then question 2.2.5, explain the effect of the unilateral light on the distribution of auxins in the plant in apparatus C. Okay, so the auxins moved away from the light so that the darker side had high concentration of auxins and the light side had a low concentration of auxins, which caused the plants to basically just bend toward the light. Right, so guys, that brings us to the end of today's presentation. Textbooks that we used for this lesson was the grade 12 Solutions for All textbook, the Mind the Gap study guide, ACID study guide, as well as Study Master Life Science study guide, which you could still get on Snaplify until the 31st of December. I thank you very much for tuning in to this lesson. If you do have any questions, please do send an email to stemdigitalschool@africateengeeks.co.za, and all the best with your final exams. Good day and goodbye.